In Griffith's experiments, Griffith used 4 mice. In the first mouse, he injected rough strain bacteria into the mouse, and it lived. In the next mouse, he injected smooth strain bacteria into the mouse, and it died. In his next mouse, he injected heat-killed smooth strain into the mouse, and it lived. Lastly, he injected a rough strain bacteria and a heat-killed smooth strain bacteria into the mouse. It died. This confirmed his suspicions that bacteria could transfer genetic information through translation.
This is DNA structure. For DNA structure, you have to have two sugar phosphate backbones. To hold the sugar phosphate backbones together, there are hydrogen bonds. Now, thymine and adenine always go together. Cytosine and guanine always go together too. So, first we'll put a thymine, and then what holds the two nucleotides together is a covalent bond. Then, adenine. Then we put them on the sugar phosphate backbone. Okay. And then let's do a cytosine and guanine. So, cytosine, the covalent bond, and the guanine. Okay. And then we'll just do another cytosine and guanine. So, cytosine. And thymine and adenine. So this is the completed DNA. Now, a regular DNA would have a double helix. So there's two lines that are twisted like this. DNA replication starts with a normal DNA strand. First, helicase unzips the DNA strand at the covalent bonds and splits the strand into two pieces. Then SSBs attach on both sides to keep the strands from reattaching. It is important to note the directions of the strands. The leading strand is 3-5 direction and the lagging strand is 5-3 direction. The lagging strand synthesizes discontinually. RNA primase lays down an RNA primer so that the DNA polymerase has somewhere to attach to. Then the DNA polymerase attaches and starts building onto the three and attaching free-floating nucleotides. On the lagging strand, polymerase synthesizes discontinue because of its 5-3 direction, so it forms Okazaki fragments which are later connected. After polymerase finishes synthesizing the strands, RNase H comes in and removes the primer. Polymerase comes back in and fills in the gaps that were left by the primer. Lastly, DNA ligase connects the Okazaki fragments. So there are two new strands of DNA.
Protein synthesis is a process in which a protein is created using mRNA. There are 2 steps to it. The first is transcription and the second is translation. This is an overview of it, which we'll come back to after talking about each step individually. Transcription. The first step of protein synthesis is transcription. In transcription, a complementary mRNA strand is created. To do so, an enzyme opens up DNA at a single gene. RNA polymerase then begins synthesizing an mRNA strand. Once completed, the mRNA falls off the DNA strand and it passes through the nucleus into the cytoplasm, and then the open gene closes. Translation. Now that we have an mRNA strand to build a protein from, we can start building the protein. A codon is a section of 3 nucleotides on an mRNA strand. This is what we read from to get to the correct amino acid. A ribosome moves along the mRNA strand, reading 1 codon at a time. Once the ribosome reads a codon, it calls for the correct tRNA with the right amino acid. These amino acids are held together by peptide bonds. The tRNA has something called the anticodon. which is what attaches to the codon. It follows normal base pair rules, except it uses uracil instead of thymine. So if the codon was GUA, the anticodon would be CAU. Although the amino acid we use is complementary to the mRNA strand and not the tRNA. The ribosome moves down the mRNA, reading one codon at a time, but it takes up 3 codon spaces. This means that it holds in 3 tRNAs at once, so once a tRNA is no longer being held in by the ribosome, it falls off. Once the mRNA is fully read and the protein is completed, the protein folds and the mRNA is deleted.